Antibiotic resistance we normally associate with hospitals, clinics, or the health sector in general. But did you know that antibiotic resistance is also becoming increasingly problematic for our environment? Today we speak to Professor Michael Gillings from Macquarie University. Michael is researching antibiotic resistance for many decades already and is giving us a brief overview about its origins and why it might become problematic for the environment. Michael, what is antibiotic resistance?
Okay, so antibiotic resistance is one of the key medical crises facing humans and agriculture in the 21st century. It is actually one of the best examples of ecology and evolution in action, and it's one of the reasons why understanding how ecology and evolution works is so important for managing the modern world. To understand what I mean, we should go back to first principles and ask, what are antibiotics? Antibiotics are members of a whole group of small molecules that bacteria produce. So bacteria live in complex communities and they need to communicate with other cells of their own and of different species. And to do that, they produce a whole series of small molecules. Those small molecules are released from the bacterial cell membrane into the environment and diffuse away from the cell that produces them. If we screen those small molecules, some of them, a very small proportion of them, if we concentrate them up and use them in much, much larger concentrations than are ever found in natural environments, actually kill bacterial cells or inhibit their growth, but don't damage our cells because our biochemistry is different from the biochemistry of bacterial cells. So think about this. You've got a cell. It's producing a whole series of small molecules. Those molecules have to then make their way into the environment. How do they do that? Well, there has to be a protein pump in the bacterial membrane, or it's a pump made of protein that pumps molecules out. So right there, we've already got antibiotic resistance because the cell that produces the small molecule has to be able to pump the small molecule out into the environment. And that means that cells that produce the antibiotics that we use already have to be resistant to the antibiotic that they produce. The other thing is that all of those small molecules are part of biochemical pathways. A goes to B, goes to C, goes to D. If C is the small molecule that's being pumped out into the environment, then the bacterium already has an enzyme that degrades C into D. And so there's antibiotic resistance as well. Already before humans appeared, bacteria are producing all sorts of small molecules that they're releasing into the environment to communicate with each other and to warn each other that, hey, I'm here, don't grow over here. There are two schools of thought about these small molecules. One is that they're signaling molecules, communication molecules. The other is that a proportion of those molecules are actually used for bacterial warfare. Now, I'm firmly in the first camp because the ability to produce antibiotics and release them is subject to, the cube rule of concentration gradients. So if you're a bacterial cell, you're small. How do you produce enough compound to diffuse away far enough and still remain at a high enough concentration that you're going to kill your competitors? And the answer is you can't. It's only when humans come along and what we do is we find a compound and we make it at huge concentrations and give it to animals or patients at such high concentrations that it becomes lethal or inhibitory for the bacteria that we're trying to control. So this whole notion of antibiotics is really an anthropocentric one. They're just small molecules that we have co-opted to control bacterial growth. Okay, so here's where ecology and evolution become important. We understand how these molecules are being made. We understand how they're transported into the environment. But bacteria work a bit differently from animals and plants. Their genomes, their DNA, are quite fluid. And what they can do is they can acquire genes from other organisms. So if you're an antibiotic producing cell, you already have the biochemistry to degrade the antibiotic. You already have the efflux pumps in your membrane to pump the antibiotic out of your cell. If a medically important pathogen can steal the genes for either of those functions, it becomes antibiotic resistant. Either it can degrade the antibiotic or it can pump the antibiotic out of its cells quickly enough so that the antibiotic doesn't have an effect. So that's called lateral gene transfer, and it's one of the major ways that bacteria make evolutionary steps forward. And that in turn is dependent on the ecology of bacteria. So this kind of process is most likely to occur where cells are undergoing some kind of stress and when they're in close proximity. So think about this. We give animals and humans large quantities of antibiotics, which goes into the gut, where there are 10 to the 9 cells per cubic centimetre of all sorts of different species. And those cells are now under stress. That turns on a whole bunch of biochemistry in bacterial cells that makes them more permeable to DNA from other cells. And so the very place that you're trying to treat is actually the ecological location where most of this gene acquisition will take place. And so the way that we administer antibiotics and the circumstances under which we do so make it more likely that the target cells we're interested in controlling are going to acquire these new genes. And that will make them antibiotic resistant. Because of that circumstance, every single antibiotic that has ever been developed, resistance has appeared within about a decade. That's just a consequence of natural selection and evolution in action and the ecological circumstances under which we use antibiotics. There is a second process that goes on here. Because bacteria swap DNA, there are DNA elements that parasitize that activity. They're called plasmids and transposons, and they're mobile DNA elements. And they make their living by moving between cells. If one of those mobile DNA elements can acquire an antibiotic resistance gene, it gives them a huge selective advantage when they get to a new species, because that species is now resistant to antibiotics. It propagates and increases in population, and at the same time, so does the mobile element. So these DNA parasitic elements, selfish DNA elements, actually prosper when they get a gene for antibiotic resistance. A really good example is resistance to colistin. Colistin's a real old school antibiotic, and there was never a problem with colistin resistance, except in a few cells that had mutations for colistin resistance, and we knew what species they were and they were controllable. About a decade ago, the colistin resistance gene moved onto a mobile DNA element, a plasmid, and within about 6 months, that plasmid had spread throughout the world because it had such a huge advantage anytime colistin was used, and because the world is now so interconnected. All of our agricultural produce, all of our animals, the pollution of waterways, all of those things are in ballast, water ballast in ships. The world is completely connected. And so it spread within 6 months to every single continent except probably Antarctica. And that gives you an idea about how environment, ecology, and evolution are interconnected, and that all of these things act to spread and enhance antibiotic resistance, largely driven, of course, by human activity, both humans taking antibiotics and also giving antibiotics to agricultural animals.
So when we think about antibiotic resistance, as you also mentioned, we often think about the health sector where it's extremely dangerous.
Yep.
However, it also has an environmental issue.
Mm-hmm.
Can you expand on this a bit more?
So one of the realizations that people have come to, it's now called One Health, which says that the animals, agricultural, companion, and other animals, wild animals and wild environments, and human environments are all connected, and that genes flow through and bacteria flow through all of these different environments. And so one of the problems here is that standard medical curricula don't actually include a lot of ecology or evolutionary studies. For instance, medical journals, they talk about the emergence of antibiotic resistance as if it were somehow sitting in a corner doing nothing very much, and then it kind of appeared magically. Whereas in more environmental or ecological or evolutionary papers, they always talk about the evolution of antibiotic resistance or the selection of antibiotic resistance. And that fundamental disconnect between understanding ecology and evolution and the practice of medicine is one of the things that drives the spread of antibiotic resistance. So I would like to see a much more evolutionary approach to medicine that incorporates environmental aspects. For instance, one of the most interesting places for not only antibiotic resistance, but biocide resistance, metal resistance, resistance to bacteriophage, things like that, is actually human wastewater plants. Because we take all of the human microbiota, we put it in a pipe, it's full of nutrients as well, we shove it into a wastewater plant where there are huge numbers of bacterial cells of all sorts of different species, and it's a giant soup for exchanging DNA. And it mixes the DNA of all of the individuals and animals in a catchment. And then you release it into the environment. And of course, sewage systems or waste treatment plants are designed to remove pathogens and bacteria, not to remove DNA. We looked at estuaries along 4,000 kilometers of Chinese coastline, and every single estuary had hundreds of millions of antibiotic resistance genes that we know came from either human guts or animal guts. Every single estuary per cubic centimeter. Enormous numbers of these genes, which were not there 70 years ago. They just weren't at anything like that abundance. So unfortunately, too late to close the stable door, right? These genes are now at enormous abundance everywhere. You and I both have plasmids, integrons, antibiotic resistance genes in our gut as permanent residents of microbiota in our gut. There's no way we can get rid of them. And so it's here for good. One of the reasons why is also an ecological reason, because if you've got a sensitive population and you expose it to antibiotics, if there's one resistant cell there, it suddenly goes to 100% of the population. And so you get this shape that goes nothing, 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 100%, but then the half-life drops down like this, really, really slowly, because there is a cost to having antibiotic resistance genes, but it's not a huge cost. So they decline very, very slowly in populations. And the chance of no one being treated with that antibiotic for the amount of time that it would take for the antibiotic resistance gene to go to zero is zero, because most people have like one antibiotic course a year, for instance. So, and still around the world, lots of countries use antibiotics routinely for animal production. We don't do that in Australia, but it's still really common elsewhere in the world.
So new molecular tools are developing rapidly. What do you think will be the next major steps for the future?
That's a really good question. And anytime anyone answers that question, they're always wrong, because developments are so quick now. When I first started in the DNA business, because I'm a DNA person, you could get a paper in Nature or Science for sequencing a small gene. That is now trivial. In fact, when I first started doing DNA work, this is a DNA sequencing gel. This is real old technology, and what you had to do was say, read this off, GA, 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 and you would type that in manually into a word processing program. And you can see that just to get 8 samples, you have to run these huge gels and then type each one of these sequences in. Now, we've moved towards DNA sequencing being routine. So, for instance, this is a DNA sequencer. And whereas this thing here would have taken me maybe a week to get, I'm going to say about 800 or 900 nucleotide sequences in the probably, I don't know, mid-90s, 1990s. This thing here is a modern nanopore DNA sequencing unit. You can get 2.7 million sequences, each of about 1,000 base pairs in 5 hours. And that just breaks my mind. And it means that, you get terabytes of information. There's the major stumbling block is now analyzing the amount of information that these things produce. So we are heading towards a situation where ecology will involve going out into the field and having these things, which run off a USB, preparing the DNA in the field and just sequencing everything. So you don't have to survey, you just go and grind everything up and put it through this, and it says, oh, here's the species list and here's the abundances.
Michael, you're highly active in your academic career still, and that for many, many decades already. Could you share, then probably also in the light of these recent technological advances and so on, any advice for early career researchers or students interested in academia?
Okay, so you don't do academia to get famous or rich. You do it because you're interested in stuff. And so my advice to people would be, do what you're interested in. Don't get stuck in one thing. Try and reinvent yourself every 5 years or so, because science moves quickly. And you don't want to be doing the same thing in 20 years' time as you did for your PhD. You want to be doing something new. And the third thing, and I can't stress this enough, work with people who are better than you. Try and find people who are cleverer, more productive, more interesting than you are, and work with them. Make sure that you do work with them, though. Don't just sit on their coattails.
Thank you so much, Michael.
No worries. Pleasure.